Thank you. I am sorry I'm a little bit late. So we have some computer problems, but I think it's not just me. I want to thank all the people who invited me, and this actually is not the talk I was originally invited for. This is tomorrow, but they asked me to make an extra talk. I hope it will be of some interest because there's a very diverse group of presentations today. I hope I am not speaking too fast. I am from London, but I live in New York a long time, and we speak very fast in both cities. So I'm going to talk this afternoon about some things we are doing with liver. It's very basic science, so I know that maybe not everybody is here for the basic science, but I try to give you something. I hope it's interesting. So my work is about calcium. And I don't know if you can see, but this is a calcium, but it's for people who are used to the Japanese art, the beach, and so on. It's Ca and 2 pluses. So this is the logo of my lab. It's a wave of calcium. So I wanted to tell you why we study calcium a little bit. So in the liver, we're interested in the endocrine regulation of metabolism. And so the hormones we're mostly working on are like glucagon and norepinephrine. This is nitric oxide. We also use vasopressin a lot. It's a stress hormone for liver. And of course, the antagonist to these hormones is insulin. And I'm interested in calcium, the ion, as a signal. And in the liver, it's a primary signal for several of these hormones. And it works through this signaling pathway that we call IP3. And I don't know how familiar everybody is, but I'm going to just quickly explain what is that. So, of course, the calcium signals then affect glycogen metabolism, they affect gluconeogenesis, they affect fatty acid oxidation, and I include here the mitochondria because we have been very interested in the way in which calcium signals, which are generated by some of these hormones, affect mitochondrial metabolism. And I just add to this, so these are some of the normal signaling pathways and some of the pathophysiologic pathways that I will talk just a little bit about. For example, some of these hormones that work through stress and inflammation, they can overstimulate the pathway. And when the pathway is overstimulated, has a number of effects, but one of the most important is to either excite the mitochondria, and this can lead to oxidative stress and generation of reactive oxygen. So, too many devices here. So I'm not going to talk about this slide, but I just want to tell you what we do. We use chemical fluorescent dyes that we can load into living cells to measure calcium. So we're measuring the calcium in the cytoplasm of cells. One of the dyes is this thing called Fura2. I'm not going to explain the whole slide, there's not enough time, but it's used to load in a living cell, and it allows us to measure the free calcium in the cytoplasm. And another are genetically encoded calcium sensors, and these are made from the molecule calmodulin that you may be familiar with as a calcium sensor in cells in many tissues, and regulator of kinases. And it's coupled to these fluorescent proteins that come from the jellyfish, the green fluorescent protein and other colors actually. And we can put this in different parts of the cell, so it allows us to measure calcium with genetic encoded indicators that are expressed in the cells. I'm going to talk to you about this signaling pathway. So the basic idea is there are G protein coupled receptors. They couple to a phospholipase that breaks down this phospholipid. It's called phosphatidylinositol bisphosphate, or PIP2. And it makes this second messenger called IP3. It also makes diacylglycerol. And these are signaling molecules for the liver. And the IP3 acts on a calcium release channel inside the endoplasmic reticulum. And also the calcium, together with diacylglycerol, activates this very important group of protein kinases called protein kinase C. And what I want to tell you is a little story about the complexity of the calcium signals and how they are translated into metabolic regulation. So you would expect from a signal pathway like this that when the hormone, this is norepinephrine, binds to the receptor, it should cause IP3 to be made, and it should activate this signal pathway. So the calcium signal should be proportional to the occupation of the receptor. And while the receptor is there, or while the hormone is there, should be always on. But in fact, we find something different. And we found, this is some years ago, that the calcium signals when, this is alpha adrenergic agonist phenylephrine, when the calcium signal is permanently on, when the hormone is permanently present, the calcium signal is not continuous but is discontinuous. It makes something, it looks a little bit like action potentials or cardiac calcium transients. And these calcium transients are generated somehow by the signaling pathway. I have to go backwards. Oops. By this signaling pathway. And so we're interested to understand how that happens. And why is it important? Because if you look at these calcium signals, they're very discrete. They're very separated from the baseline of calcium. And if I show you these red traces are the integrated calcium. It means what would happen if that system worked just proportionally to the hormone activation of the receptor. And you can see these calcium signals that come, that would be the integral of these, are very small changes. So this is, if you think the way a nervous system works, it works with frequency of action potentials. It does not work with the amplitude. And this is a similar system for calcium signaling in the liver. And basically, as you increase the hormone concentration, the frequency of this calcium spiking is increased. So we think this works, we know this works actually, by a process that involves feedback. So this is the signaling pathway, and the calcium that's released by the channel is activating the channel. So this gives rise, I show you this. This is a plot of the calcium. So this positive feedback causes a very sharp calcium increase. So anytime this signal is activated a little bit, it's activated a lot. And then, because also there is a negative feedback of calcium, all on this one ion channel, the negative feedback makes the calcium go back down. But these have very different time courses, time constants. This is fast and this is slow. And so you get a process of repeating calcium spikes, and the only thing that determines the frequency of these spikes is how much of the hormone, how much IP3 is made, that determines the relative rates of the on and the off reaction. So I'm going to, we'll see if this works. So these calcium signals are actually, the red, these are individual hepatocytes. They are loaded with one of these fluorescent dyes. This is how we make the measurements. And you can see these calcium signals, they are not they're not completely in the cell all at once. They actually make a kind of calcium wave. So this is one cell, and you're seeing the calcium. Here's another one. And these cells are in culture, so they're not connected to each other, which means that they're all asynchronous. And so I just showed you there are a few. So the calcium oscillations are actually waves of calcium. And I'm going to show you how that works in a minute. But I want first to show you that we can make the calcium oscillations even without the hormone. So in this experiment, I'm showing you a diagram. We use I have to go back, I think. Excuse me a second. Okay, so we take this compound, this second messenger, IP3, and we have it chemically caged, which means it's not active, but it can be activated by a flash of laser light. So if I go forward, so in this is the chemically caged form, which also can be loaded into cells. So when we have this in the cell, if we flash on a UV pulse of light, it will release the active second messenger. So we can ask the question, if we just put the second messenger on, do these cells give still the same kind of calcium signals? And this is it's working, good. So this is an image where we're doing that. And in a few seconds, I think already there was one, you will see a flash of light, and then again there. So this is the calcium signal that we cause by releasing IP3 inside the cells. And you can see now they're quite active, and then there's another flash. It makes more IP3, and you begin to see these things. But you'll see they're very local, calcium flashing in different parts of the cell. Trying to make a wave like the one I showed you with the hormone, but they're not able to do it completely. So and I'm going to be one more flash, I think, coming now. even with the highest level of IP3. But now we put on, this is vasopressin, and you will see something very different. The hormone makes these calcium waves you'll see in this cell. It goes all the way through the cell. And again, and this is in the continuous presence of the hormone. And again, down, I think this one does it, this one is doing it, this one, this one. So I'm going to show you, I'm going to move on and show you the trace. So if we look at the trace of the cells, how is the calcium signal? This is one of those cells. You see here, each one of these is releasing the IP3, the second messenger, and with each pulse, we see first very little, and then 1 pulse, 2 pulse, 3 pulse. And what we did is we measured in 2 parts of the cell. So there is a pole of the cell, it's the apical pole, the secretory pole of the hepatocyte, where the calcium signals begin first. And you see here that these calcium signals at low levels of IP3, they're very local. They only occur in one part of the cell. And as we increase the signal, we eventually get them to appear all over the cell with the maximum IP3. But when we put on the hormone, we still see something different, because from these very short, brief releases of calcium, we see a broad, wide calcium signal. So I want to tell you how that comes. You will tell me if I'm going too long. Okay. All right, so I'm going to tell you how that comes. So it turns out that the calcium signals that are made by IP3 are just by the mechanism that I showed you before. And I apologize, I keep having to go backwards because I didn't. So the calcium signals made by uncaging, by releasing IP3, are made only by this mechanism. But the calcium signals with the hormone involve another step of calcium, and that is calcium acts on this enzyme. So there is another positive feedback, and I want to show you the evidence for that. So here is a measurement of, in the top panel, calcium. Again, you see this continuous application of hormone and periodic oscillation of calcium. And here we measured the IP3, and we measure the IP3 using another genetic indicator. It's part of the IP3 receptor, I will show it to you in a minute, part of the channel, and GFP, and it's designed to cause changes in fluorescence. So you can see here the calcium and the IP3 change together. But the question, which one is coming first? Is the calcium necessary for the IP3 or the other way? So we can address that question by making a buffer of IP3, and I'll show you what I mean. So this is the signaling pathway, a little bit different diagram I already presented to you. Here is the receptor, the G protein, the phospholipase C. This is the generation of the second messenger, IP3. This IP3, it acts on the calcium channel, and I showed you already that the calcium that's released, so the calcium is stored in the endoplasmic reticulum, the calcium that's released acts positively and negatively. So we know that that can cause calcium oscillations, but we also look at a couple of other places. So calcium has this positive feedback on the phospholipase C, the same enzyme, so it can increase IP3. And we also know that calcium can activate this enzyme, the kinase that removes IP3. And in fact, if you think about how this can work, because the IP3 is made with positive calcium signal and removed with positive calcium signal, this can be made to oscillate. And what we did was we created a buffer, just like a buffer of pH, that prevents the IP3 from changing. So we put this molecular buffer. I'm going to show you how it is. This is the gene for the IP3 receptor, the calcium channel, and we took just the part that binds to IP3 and we coupled it to another fluorescent protein, and we use this as a way to block the calcium changes that are induced by IP3. To block the IP3 changes, and we have a mutant that doesn't bind IP3, so this is our control. So if we put this in the cells, here you see the mutant that doesn't bind IP3, and we see this calcium spiking. And if we put the IP3 buffer, we completely remove the calcium spiking. So this then is evidence that the IP3 itself has to change to allow the calcium to change, because this buffer does not prevent, I don't have time to show you all the data, does not prevent the IP3 from increasing, it just prevents it from oscillating. And I'm going to skip, well, there's one more slide here. Yeah, I skipped the slide, it's too complicated. So what is the purpose of this? The purpose is to allow for a increased range of frequency of spiking. So this is a little complicated from a modeling study that we publish in Biophysical Journal. And if you look here, this is the rate of the PLC. This is the enzyme. So this is really the strength of hormone activation. And this is the actually we plot as period. This is the interspike period. So how fast are the oscillations going? And if you don't have that ability to feedback on the IP3 formation, that feedback on the phospholipase C, you get very small range of oscillations. But when you allow calcium to act on the phospholipase C and change its activity, you get these very big ranges. So this allows from a range of IP3 and calcium from very, very slow to very fast. And in fact, this is what we measure in the cells, and we know that this is linked to the regulation of various metabolic pathways. So if I take the slide I showed you, we add now another feedback, and by the combination of these feedbacks locally and this feedback on the receptor system, we have the ability to create these very long calcium cycles, oscillation cycles. So if we look back to I showed you a few minutes ago the waves of calcium in a single cell. So if we look at how that seems to work, the calcium is released from these IP3 channels with calcium in the endoplasmic reticulum. And these calcium releases, we call them sparks and puffs, that are made by IP3 alone. When the phospholipase C can be activated by calcium, they transmit through the cell. So you get a wave of calcium passing through the cell. And if we extrapolate that to the intact liver, we know the liver cells are coupled by gap junctions. And so what we expect is those calcium waves can go from one cell to the next cell. So I'm going to show you data from a preparation of intact liver. So we take this is all with rat liver. We take the liver in a perfusion. We perfuse through the portal vein, and we can introduce into that portal vein any hormone we need to add. And we can look then in a confocal microscope that allows us to look deep inside the tissue. We can look at the calcium signal organization. So this is very low magnification. You're probably familiar with the structure of the liver. So this is the portal tract, this is the central vein, and there's these plates of hepatocytes which have also secretion of bile into the canaliculi. And this structure here, this is actually central vein. So this is this. This is the size of the laser dot. The dot I show you is about the size of one cell. So there are many thousands of cells. This is close to 1 millimeter of the liver. And it's not the surface of the liver, it's inside the liver by optical imaging. So if it works, I can play it. So what you're going to see here is when we introduce the hormone, the calcium, the red is where calcium is increasing. The calcium is increasing and spreading out, and we're continuously infusing the hormone, but you see the discontinuous signal. Calcium is increasing, it spreads out. These are 2 hepatic lobules, and you'll see a third one continuing to spread through the tissue. And I'm going to go to the next one and just show you. So this is now at high magnification, and if I can find the mice, here we go. So in this case now, one cell is about this size, and you're going to see this cell is the first cell, and these cells are passing these calcium signals one to the next to the next. And the function of this is it allows for synchronization of, for example, glucose output from the liver. It allows for it allows for detection of a local signal and propagation through the liver. So I want to show you, this is one more experiment. This is the experiment to show you this is due to gap junctions. So these are running together. In this side is the control, and in this side we use a blocker of gap junctions. And you see, whereas this one, the calcium spreads out as a wave, in this one they are all asynchronous. Individual cells are responding, but they don't couple to each other. So this is our evidence for the gap junction connection. And in this experiment, in this slide, I'm trying to explain how that's working, that the gap junctions between the cells allow this signal of calcium and IP3. The calcium is regenerating IP3 across the cell, is passing through to the next cell, and so you get this positive feedback between calcium and IP3 that allows propagation of these signals over very long distances. So the last thing about this, this preparation is we can, instead of infusing the hormone, we can try to release it locally with nerve stimulation. So we're going to, this is another picture of the liver, and I'm just remind you that the liver is innervated with sympathetic, actually also parasympathetic, but we can electrically stimulate the nerves going into the liver, and I'm going to show you only one example of this. So let's see if this one will play. So in this case, we're stimulating the sympathetic nerves, and you're going to see that there's an area that's innervated. And although the nerve terminals are releasing only by 1 or 2 cells, these calcium signals are again spreading through many cells. So it's an amplification of the signal that's detected very locally. I don't know how I'm doing for time. So I'm going to give you one more example of how we use these imaging technologies. This is work with a collaborator at San Pablo University in Spain. And Fernando is interested in metastases and how they locate with us in the liver. We're interested in other tissues. So we're trying to use our intact perfused liver system to look at what are the changes that occur on first contact of a metastasizing cancer cell. So a cell which has come, we're interested particularly in colon cancer as one which is commonly gives secondary metastasis to the liver. So this is a still shot to show you a little bit how it works. So this is in fact intact perfused liver, and now I showed you before the hepatocytes. This is staining with a wheat germ agglutinin to just stain the endothelium. So the hepatocytes, actually you don't see them, they're here. This is the lumen of this is the sinusoid lumen. This is the endothelium, and the hepatocytes are invisible because they're not stained. And this is one of the cancer cells. And what we do is we pre-label the cancer cells by making them express a green fluorescent protein. So in this case, the green fluorescent protein is, the cancer cell with the green protein has been injected into the portal vein along with the perfusion. And this is an example showing how they arrive in the liver, but I can show you the movie, it's more interesting. So, I hope. There we go. So in this movie, these are now, you see the hepatocytes. They're labeled with our calcium dye, actually. But what I'm going to show you first is just the arrival of the metastasizing cells. I'm trying. Oh, here we go. So you will see these cells, they arrive, and they find a place, they seem to stick, and I don't have time to show you all the data, but the sticking is due to protein-protein interactions with specific endothelial cell markers. And if we go to the next slide, so now I show you the calcium part of the signal. So again, as with the other images, this is in red. And so you'll see, there you go. So you'll see the cancer cells arrive, and then a little bit after that, you begin to see some calcium signals, and they're calcium signals in the hepatocytes. And what we think is those calcium signals in the hepatocytes are responsible for allowing the invasion, allowing a reorganization of the cells. So I don't know, you see there's one or two here. And they're only in very few cells. They're just in the ones in, not just in contact, but in nearby cells. So if I go, this is a still shot just to show you the same thing. You can see there's about 5 or 6 cells here affected by a cell that arrived here. And the last slide for this is to show you that the way we think this works. So the cancer cell is first contacting the endothelium, and the endothelium is generating nitric oxide as a signal, which I didn't show you the data, but we have shown before is affecting the calcium signaling. So this calcium signaling by nitric oxide uses cyclic GMP, acts on this IP3 receptor, and this calcium signal allows for rearrangement and reprogramming of the genes of the hepatocytes. I think I ran out of time. I have one more thing I could show you, but I think I will stop because there are many speakers and we started late. So I will just tell you that the last thing you should know is these calcium signals also control mitochondrial metabolism, and I'm going to skip by these slides, but I'm going to give you the summary. So we're also able to measure the calcium in the mitochondria, and we can look at the metabolic responses. And the important take-home message from this is these calcium oscillations translate to mitochondrial metabolic oscillations, But if the calcium signal, whoops, one more. If the calcium signal is too high, the calcium generates overproduction of activation of the respiratory chain of the mitochondria and generates huge amounts of reactive oxygen because the respiratory chain is overloaded. So in this experiment, we put a high level of hormone that causes a sustained calcium increase instead of these transients that we usually see. And now you see with the sustained calcium increase, this large generation. This is superoxide, again measured with a genetically encoded fluorescent protein probe, so not a chemical probe. So I think with that, I will, there's another movie, but I will finish. So these are the people in my lab in Newark, in Rutgers, that did the work. Most important, Larry Gaspers, Stephanie, and let me see who else did this, and Paula. So I think those are the ones who were there who did this. So I thank you. And